Hey, welcome. So this is a video companion to my What is Aging essay, but with more graphics and maybe some drawings. Um, so basically here I wanted to go over some of the same topics and just expand a little bit on them, perhaps from a slightly different angle. Um, the reason I wrote the What is Aging article in the first place is that if you read various places that talk about aging, you don't find a definition of what is aging. People will talk about the aging process of aging is causing this or aging is causing that, but what is what is aging itself? For example, if we start by my longevity FAQ, at some point here is I don't define aging in the longevity FAQ. I just say that, well, as time goes by, mortality risk increases. Is that aging? No, it's just the description of the fact that as we chronologically age, we are around for longer, the odds that we won't be around for longer decrease, which is the compared Mayhem law. And then in what we age, I more or less gesture towards a view of something about damage accumulation and damage repair and an imbalance between those things. And there is something to that, but it's an incomplete view. But it's a view that is directionally correct. For example, the view that the Sense Foundation made popular, so Sense will stress that all there is to aging is the unrepaired damage to cellular and molecular structures that make up the functional units of our tissues. That is a damage accumulation. And so Sense will then say, well, that damage is these various categories of damage. Now, Sense doesn't claim in this case that these are the root causes of aging. They have never claimed so. They just say that, and actually here you can see that ultimately what leads to that we don't really know. But then they say we don't have to know. That in their case, it's like, well, we can just But what they claim is that if we fix those things, then we can cure aging, which is quite nice. But still, if we just get to the what is aging question again, the idea here is that aging is seemingly, to some extent, some form of damage accumulation. If we go to another source, for example, Laura Deming's longevity FAQ, which is the OG longevity FAQ, if I wrote mine, we start with the observation that, again, as in the one I wrote, that as we age, the odds that we die increase, and so does the odds of contracting any kind of age-related disease. Why this happened? Well, again, some pointer to damage accumulation, as is typical in aging explanations, and some pointer to we don't really know what's going on. And then we move from what is aging, which is more or less gesture, to what is the study of aging, and then we find various things. We find, again, a focus on damage. What kinds of damage are those? How do we reverse damage? And what switches could we flip in human biology to increase lifespan? So this would be either from the point of view of a gene therapy or gene editing, what could you do to increase lifespan, which in the context of the explanation, it would be how do we decrease the rate of damage accumulation, or how do we, if there's something we can flip to then reverse that damage accumulation. We could also go, for example, to the hallmarks of aging paper. First we have the from afar the American Federation for Aging Research, where they will point to the hallmarks of aging. So they will say, aging is a bunch of interconnected processes. Okay, well, that's kind of sounds a bit vague, but again, it's kind of directionally correct. Then something here I would disagree with them in that they say that there are some hallmarks which are at the core of what aging is, and that's kind of unclear, but the hallmarks, they, as people repeat over and over, the hallmarks were never meant to be root causes of aging. They were just a collection of things. If we just go to the actual paper, if we go to the paper, what they actually say, well, first they have this is their definition of aging, and time-dependent functional decline that affects most living organisms. Again, this is not a good definition because, well, everything's time dependent, right? You could take any process and say, well, it has to be time dependent. I mean, aging is change, and change requires time. So it's not really telling us much, and it's not telling us what the nature of the process is. But in any case, the definition of a hallmark is this. This is what a hallmark is supposed to be here. So hallmarks are things that happen during natural aging, things that if you make them worse, aging gets worse, and if you recover them, then aging gets better. Again, no mention of root causes here, no mention of key drivers, just things that go with aging. So you could add plausibly things like the increase in LDL cholesterol with age, right? Because it goes up with age. If you add LDL cholesterol, increased risk of death. If you decrease it, then you decrease the risk of death. Here, that LDL analogy then gets us to a different point, which is the reason why the damage accumulation viewpoint is on its own incomplete is that you could take someone that is, let's say, 18 years old, let's say, and then you plug into your veins, just basically inject them with LDL cholesterol, lots of it every day. They will probably die quite soon. They will develop a cardiovascular disease very early. Have they died of aging? Well, they certainly died of damage accumulation, right? They have accumulated plaque in their arteries, but we won't see that as aging, even though it's exactly the same damage accumulation that we see in aging. And in someone that dies of heart disease at 50 or 60 or 70, it's because they have accumulated the exact same damage that the young person accumulated in this thought experiment. So to me, this is why in the essay I argue that we have to supplement this damage accumulation view with the idea that aging, there is a systemic component to aging. The idea that many processes are degrading concurrently and with some positive or negative synergies going on between them. Which is why I also say that aging does not have to do with time. As I said, well, obviously you need time for any change to occur, but you could plausibly cause or you could rather accelerate or decelerate aging. It's not given by how long you're alive, or to put it in a different way, two people that live to the age of 50 or 60, they can age at different rates. So it's not so time is not dictating the speed at which aging proceeds. It's driven by other processes, which sure the time what time is doing is literally, for again to go back to the case of cardiovascular disease, let's say there's an LDL cholesterol particle and there is some chance it will stick to an artery, but there is some chance it will not. Now with age, you're just buying kind of lottery tickets for various kinds of disease, and it's just because you're buying more of them that time correlates with disease and seems to lead to aging. But they wouldn't say that time causes aging. And as I argue in the essay, the cause of aging is to some extent noise or entropy, plus some potential degree of quasi-program aging patterns, which the extent to which it's just noise or it's just this quasi-program aging stuff, it can be debated, and it's probably a species dependent. But it's either it's noise, unintended noise, or is something in the system that is actively pushing the system out of the healthy state to which the departure from which we consider to be aging. Aging also depends on the organism. So here there is this very interesting paper from Adam Freund from Calico, and he has this diagram here where he says that there might be different aging processes that may or may not overlap. At least that maybe they each, if to treat different diseases, maybe you have to treat them separately, or maybe you have to treat them together. Or to put it in a different term, there is this sort of picture where we have various kinds of aging phenotypes. This could be Alzheimer's, wrinkles, or various sort of things, and different causes. And in one case, each of these has their own cause. In the other extreme, there is a single cause. And in the middle, we have a set of common causes, which is also my view that, and perhaps the so-called neuroscience hypothesis, which is that treating a smaller number of causes can then help you treat a bunch of diseases. That is true. But the extent to which it's true, you could argue endlessly about that. For example, if you take and especially when you take the point of view of developing therapy. So if you take, for example, senescent cells, right? So you could argue that senescent cells are having an effect on arthritis or osteoarthritis and macular degeneration in the eye and brain disease and whatever. The therapies you're going to use to tackle these might be different, which means that sure you are targeting senescent cells, but you're targeting senescent cells in different places. Maybe the kind of senescent cell that you're targeting is different. So because there are various kinds of senescent cells, so maybe you ultimately end up developing completely different classes of drugs. So one could say, for example, let's go to this. One could say if you have, for example, let's see if I can get this to work. Okay, if we have Alzheimer's and wrinkles and cancer and diabetes and fatty liver and like white hair, what else do we have here with aging? Well, I guess we have increased susceptibility to infectious disease. Then we also have various kinds of pulmonary diseases, as well as, I say Alzheimer, but I could just say dementia in general. We could also point to sarcopenia, right? Like loss of muscle mass and muscle function, reduction in bone density. And we could say, okay, well, suppose we treat each of these one by one. We cure Alzheimer's, and then we cure cancer, and then so on. If we say that, have we then cured aging? Seemingly, yes. I mean, if we say that aging has symptoms, which are all of these age-related diseases, and we cure all of them, then we have cured aging, seemingly. So if we say that this is aging, which I'm not saying that the aging is something that this might be symptoms of aging, then just going one by one just means that you cure aging. But the idea or the optic of aging science is that all of these things are connected, and that you may be able to solve many of these at once with a single therapy, or that you may have to intervene conversely on various points to solve one of them. For example, take the immune system. Again, the extent to which this particular aspect of the immune system that I'm going to describe now is relevant to aging is, again, under some dispute, and I wrote an article about that you can check out on Nintil if you search for immunosenescence. So in our bones, in bone marrow, there are hematopoietic stem cells. These stem cells generate all sorts of kind of cells in our blood, and in particular, white blood cells are the ones of interest. HSCs that are going to make white blood cells, they can differentiate into the lymphoid or myeloid lineage. So they can be, let's say in this case, lymphoid is one of them. And from the lymphoid lineage, we get, for example, T cells. Now, T cells have to go to the thymus to get trained and to work as T cells. Now, the thymus, with age, can we get some color here? Yeah. So with age, whoops, maybe that was not intended. Oh no. We will have to go back to white, I guess. Yeah, it's probably here is what we want. So the thymus shrinks or becomes fat with age and it stops working. So by the time you are older, you are left with a number of T cells, and that's all you have. You're not getting any new ones. And that's might be a bad thing. Why? Well, because, well, actually, these are not T cells. These are thymocytes. From the thymus, you get the T cells. Now, these T cells might be doing various things. For example, they are fighting cancer or senescent cells or infections. suppose, for example, that we also happen to know that the brain, there are nerves that in the brain that are going into our bones, into the bone marrow, and that it seems that there are some papers suggesting that this activity may actually be directly controlling the HSCs. Suppose this is true. In that case, if we if brain function is in some sense impaired by infections or by senescent cells, inflammation or whatever, or tumors, then you may see a degradation in the immune system. So ultimately we come full circle here. That's what's the point. So we have this is one potential hypothetical feedback loop that you might find in aging. There is a paper that if you search on my Twitter for the bagel of doom, this is the bagel of doom. And this is the idea that you can have these nasty feedback loops going on in various aging-related processes. But the point of all of this is basically saying that you could, to some extent, help the immune system by targeting the brain, or you could help the brain by targeting the immune system, or you could help. Maybe we can another example here. Can we just delete all of that stuff? Yes, we can. You may have seen NAD. So the fact that NAD goes down with age. NAD is a metabolite used to basically move electrons around. So in that sense, it's like ATP, that it's like a currency of sorts. So most of the NAD is made by a so-called salvage pathway. So if you're a cell, you're just using and recycling NAD. No problem. But what if you cannot make enough NAD, or for some reason there isn't enough? Well, in this case, the liver can make a precursor to NAD, which then gets pumped into the blood, and then from the blood it goes to various cells. So that's nice, right? We can make more NAD. But what if the liver function? What if liver function goes down? Well, in that case, NAD might go down. And actually, there's a very interesting paper from the Covarrubias lab that shows that senescent cells cause inflammation. Inflammation causes the activation of N1 macrophages. These macrophages have an enzyme that's called CD38. Whoops, I think you cannot see that. There, CD38. CD38 is an NADase, or an enzyme that takes NAD and makes it into something else. So basically, the activity of CD38 goes up by a lot when in these macrophages when there is inflammation. There is some speculation about why that might be. So basically, you have CD38 that takes NAD and it makes it into something else, which I now don't remember, but that might have anti-inflammatory properties. But in any case, it's causing NAD to go down. Be that as it may, if this might be a cause why, let's say, that cells are not getting enough of NAD. Of course, it's not the only cause. Like ideally, if the salvage pathway is also breaking down, if you could fix that, then you would need that extra NAD coming from NAD precursors coming from the liver, right? But again, this is the point of aging, that in a typical disease you may be looking at the brain or the liver or the pancreas, but actually you need to look at everything and to what else that might be connected to, which was the point of my of the picture that is on my post. I think I have one at the end of the. Will this work? Can I get this to work? Apparently not. Well, apparently not. Need to learn how to use this thing. So, yeah. So, what then? So we have seen. Oh yes, something I forgot to mention before, which is that if we agree with this and we agree with that aging depends on the species, studying aging in one model animal may not be helpful to understand aging in other. In aging research, people typically will look at yeast. These are the usual models: yeast, flies, and this is Drosophila melanogaster, C. elegans, which is a worm, which we can like paint here like a happy worm there, and mice. It's the usual. And then there are some other models like that are more obscure, like killifish and like rats, which are more common. And you could look at like various kinds of rhesus, macaques, and stuff. But these models are, I mean, if you read my longevity FAQ, you see a bunch of differences that these models have. So take, for example, C. elegans. Why do C. elegans worms die? Well, there's a paper on that, interestingly enough. And C. elegans, they do for two reasons. If you look at this, if you analyze a bunch of them and you look at why they die, and you can decompose their these survival curves into two different deaths, what they call uppercase P death and lowercase p deaths. But basically, they point to here swollen pharynx or an atrophied pharynx. And in turn, these are caused by an infection or an infestation with E. coli, which is this bacteria that the worm eats, or just by mechanical damage, kind of. Okay, well, but so C. elegans worms, they don't have like they have a fixed number of cells. They don't get senescent cells. They rarely get cancer. They don't have stem cells to replace lost cells. So this would be, to some extent, like studying aging as if everything in aging were like our teeth. Like our teeth, once they are gone, they are gone. And sure, they grow, I think, twice when we're kids, and then we get a second set, and maybe you could engineer something to get a third set or get like continuous replenishment of teeth, like in sharks. But yeah, so these worms are not dying of, let's say, of cancer, or of course they don't die of cardiovascular disease because there is no blood in them that's circulating. Which then also gets us to the point of studying aging in animals other than humans. In that, sure, if you look at the cellular level, if you just look at cells, the pathways and the various kinds of proteins, they are kind of similar, but the configuration of the cells in a whole is different. Maybe from a physics point of view, if you come to it with a physics brain and you think of the standard model of particle physics, there's a limited number of particles and a limited number of interactions, and you can study things in a very simple way and then build up from that, and then everything's kind of the same. But that reasoning doesn't work here. And sure, even in physics, you can argue that there's lots of complexity in various ways different things can get combined. So maybe that higher level insight has to be translated also into biology, if you're coming thinking from like first principles kind of physics kind of reasoning. So if you're studying, for example, like in yeast cells or worms or flies, and you're looking at individual cells, you're not going to learn many things about humans. So, for example, if you're studying, there is no amount of single cell studies you can do, no amount of sequencing, no amount of understanding pathways, understanding how a single cell works, no amount of that you can do that will help you to understand cardiovascular disease. Maybe cancer you can do at the single cell level to some extent, but that, or if you take Alzheimer's, for example, Alzheimer's disease may have different causes. One of which might be, for example, the reduced clearance of various kinds of amyloid aggregates in the brain, which may be caused by, again, for this you have to take into account the three-dimensional and structural makeup of the brain. There is this, if we go back to this, and if we draw like a brain of some kind, this looks like a weird potato, But basically, we have these ventricles here that are like pumping out cerebrospinal fluid and kind of washing up through the brain, and then that's being collected here and disposed of down into your spine, and then blood, and then excreted. So the brain is kind of washing away and cleaning up stuff. Now, if this flow gets blocked, well, problems happen. I'm not saying this is the only or even the major cause of Alzheimer's. It might be one, but I'm saying that you cannot study this mechanism by looking at a single cell. Even in an organoid, even if you're studying an organoid that represents a section or a part, a region of the brain, you would not be able to study how this affects, in this case, Alzheimer's, which is a huge challenge, right? Because if you cannot go single cell, you have to do in vivo. But then mice, for example, they don't typically get cardiovascular disease or Alzheimer's. Like, they don't live long enough for that. They mostly die of cancer. So in that case, what do? You need to grow full brains or something in vats, or you need to, which is what the field ends up doing, you need to study bodies of those that died, which is not ideal because once they are not, those are not like living brains or that are kind of fresh. They have already died. So yeah. So after all of this, you may wonder, well, then what is aging? Is this it? And so this is why I like the essays of works of David Chapman, that it seems like if you spend time reading this literature, that there is something to aging. There is something to which that you can apply these words to. But I think that aging, it's maybe most intuitively characterized as an optics, a framework, or a view with which to look at biological systems. That if you have the aging glasses on, you're looking at, again, you're thinking more of connections and than you would if you were just studying cancer or Alzheimer's in isolation. So you may be looking explicitly for this kind of common causes of various kinds of disease and trying to study how affecting something or one system may lead to changes in a different system, or how the degradation of your systems feed into each other. So that, to me, is what aging research is about. You could argue, having this answer, does it change anything? As in, does your definition of aging change as a researcher, policymaker, philanthropist, whatever? Does it change what you're going to do? Not a lot, I would say. I think having a concrete answer for what is aging is not 100% crucial. And as I say, like, if you go into literature, despite all the disagreement about the question of what is aging, the field has made lots of progress. Which, again, if you're like very philosophically inclined and you want to like have clear concepts, this is not satisfying. But ultimately, it's good news that we can make progress without having it super clear what actually we are trying to do in the aging field. Perhaps one use of a good concept of aging is when explaining the field to others. As in, coming from the outside, when you read lots of papers and talk to people and look at all the different companies and what they're doing, you more or less get the idea that aging research is what aging researchers do, and you just see what they are doing, and that's aging research. But if you want to make that more explicit, then I think this optic, I think it's helpful to explain what's going on with the aging field, as in what is that they are doing, despite the fact that they don't agree of what the object of their analysis is, or how could it be that funding Alzheimer's research is to some extent or partly funding aging research as well, because again, it's part of an aging process. So yeah, I guess the mini rant gets up to here. I hope you enjoyed it. I know, enjoy your day or something.